Hello, biologists. Let's talk about the historical basis of our modern understanding of DNA. So, first of all, we're going to explain what transformation means. What does it mean when I say the DNA was transformed? This is something we understand now, but when they named it that, I'm not sure they fully understood the mechanism by which DNA was going from one organism to another. We are going to describe the key experiments that helped identify this DNA. We're going to recreate some of these in class, and we're going to explain Chargaff's rules. Okay, let's start out with Friedrich Michener. He was a physician by profession, and he isolated phosphorus-rich chemicals from white blood cells. So what he really did was studied pus. Lots and lots of pus. So much pus. Essentially, what he isolated was just the nucleus particles. And so I am going to see if this little video will play. So here's Fred. He worked in a hospital. He collected bandages and all kinds of pus on those bandages. He added a weak alkaline base to the solution, which lysed the cells and basically precipitated out the nucleus. Right? There's some pussy bandages, put it in a salt solution, added an alkaline solution to that, and it precipitated out the cell nucleus, what we now know as the nucleus. They called it nuclein. Essentially, what he found in this is that it's high in phosphorus. Keep that in mind, because as we look at the structure of DNA, we will see that ladder, and that it is made of a phosphate background. So that's why, again, it was the nucleus particles that he isolated, and it was mostly phosphorus. So when you look at the phosphate backbone of DNA, you will see why this, in retrospect, was actually quite important. So he discovered the nuclein. Now we entered also another guy, Frederick, the Fredster, another Frederick Griffin, who was trying to find a cure for streptococcus. So he was actually just trying to find a cure for pneumonia. And he ended up being a major player in the DNA structure game, if you will. So essentially he was looking, again, the point was he was looking to cure streptococcus. And spoiler alert, he didn't do it. Penicillin did it. Anyway, it got done. So he looked at living S cells. Okay, so he has the pathogenic cells. And when he puts them in the mouse dies. He also had living R cells, which were the non-pathogenic group, right? When he puts those into the mouse is healthy. Now, when he heat kills, right, so he's autoclaving these S cells, the mouse is fine. When he mixes the heat-killed S cells and the living R cells, the mouse dies. It shouldn't. Look, happy mouse, happy mouse. When you mix these two, you should have a happy mouse. But you do not. You have a dead mouse. What is going on? What is going on is DNA was actually able to move, transform. This is the whole basis of genetics, our ability to move genes from one organism to another, from one strain to another. This is essentially what Griffith discovered. He did not know that. No one knew that for 30 years after he did it. But he knew there was what he called a transforming principle. Something is going from here to here. What is it? We have several slides and lectures before we find that out. Okay, so what is transformation really? What really does it mean when we say transform? It's the development of competence. For a bacteria to take up DNA, it must be in a competent state. So it must be able to take up DNA. Now, what's absolutely crazy about some of these scientific experiments is if he had used any other bacteria, it is not a competent bacteria. It is not able to take DNA from one to another. But he didn't. He used Streptococcus pneumoniae. And what do you know? It was able to take it up. That's crazy. It's almost like Mendel and the peas. If he'd used anything other than peas, it would not have worked out as well. Same thing with Griffith. It used practically any other bacteria. This would not have worked. But he did it. He used Streptococcus. And many others can be made competent in the lab using either electroporation or calcium chloride. He didn't need that because he had pneumonia. And when he did that, he was able to look at horizontal gene transfer. We talked about this a little bit in Intro to Biodiversity. Horizontal gene transfer is when the DNA is able to move from one cell to another. And that is what we were looking at. That's what Griffiths discovered. When he added these two together, the DNA from the heat killed was able to transform into the living. So this one's alive and this one's virulent or pathogenic. And together they kill the mouse. They have living S cells, which is just crazy. Okay, moving on to the next experiment. So now we have figured out what's in the nucleus, and we have figured out that something in that nucleus is able to transform, horizontal gene transfer. Something is able to move from one bacteria to another. The transforming principle, what we know now is DNA, but we don't know what it is. They don't know. What is this? They know what is in the nucleus, but they don't know which one of those things is actually what is causing the transforming principle. So now we go to Avery, McLeod, and McCarty, and they have narrowed it down at this point. It has to be RNA, DNA, or proteins. One of those three things. RNA, DNA, or proteins. Everyone is on team protein. Completely honest. Everyone's on team protein. Proteins they're complicated. There's a bunch of parts to them. It actually makes sense. RNA and DNA, that's insane. There's 4 letters. Like the entire alphabet of the living world is made from 4 letters. That's crazy. No one believed it. So Avery, McLeod, and McCarty basically did the same thing again, minus the mouse. Does that make sense? Okay, so the heat killed some S cells. And then what they did is they took out the proteins, right? We're just going to process of elimination and see what transforms. This is a pretty simple experiment. You could have thought of it yourself, right? Thinking back, you're like, okay, let's put everything together without the mouse and see what transforms. So they added proteinase, which killed the proteins. So in this one, we don't have any proteins. In this one, we don't have any RNA. In this one, we have DNA. And, oh my goodness, it doesn't work if there's no DNA. So when there's no DNA, right, we killed the DNA because we used DNase, an enzyme that kills DNA, and we didn't get transformation. Boom. That should have been enough. Everyone should have believed it, but they did not. Here it is again. Again, they used enzymes that specifically degraded the components. For each, they degraded the RNA, they degraded the protein, and they degraded the DNA to see which one of them prevented transformation. And what do you know? It was DNA. When they took out the DNA, when the DNA was degraded, the resulting mixture had no transformation. And this should have led to a universal conclusion that DNA was the transforming principle. But it did not. Because it's a ludicrous conclusion. And everyone's like, no, there's no way. Moving on. Look at these two. Hey, you just discovered the process that DNA is the genetic material. And this is what we get here. This is these two. And we'll talk about we'll talk about this. Okay. So these guys, this is Martha Chase and Alfred Hershey and Chase. This is known as the Hershey and Chase experiment. Only one of these people won a Nobel Prize. Can anybody guess who didn't get the Nobel Prize? The woman. Spoiler alert, they left her off the Nobel Prize. So DNA was still considered to be a crazy idea. So they used radioactivity. They used radioactivity and viruses. Okay, radioactivity and viruses. They looked at proteins with 20 different amino acids, and they said, we still think it might be DNA. And everyone's like, no way, it's got to be proteins. They went ahead and looked at it using radioactivity. What they did, Martha Chase and Alfred Hershey in 1952 sort of confirmed. Finally, the multiple evidence confirmed, yes, DNA is the genetic material. They looked at bacteriophages. These are viruses, okay? The phage is a virus that infects bacteria. The viruses typically have a simple structure. So they have a protein coat called the capsid and a nucleic acid core that contains either DNA or RNA. So the bacteriophage infects the host bacterial cell by attaching to the surface, and then it injects its nucleic acid inside the cell. This is great. If it's protein, it will be in the coat. If it's nucleic acid, it will be injected into the bacteria. This is a pretty easy way to figure it out. So essentially, they selected radioactive elements that would specifically distinguish this. They labeled the radioactive sulfur, S35, to label the protein coat. And they labeled the phosphorus, right? We know the structure of DNA. They labeled the phosphorus in the DNA to see which would actually transform, which would actually end up in the bacteria, which thing would be radioactive. Anybody know? Spoiler alert, spoiler alert. It's DNA, right? So here they labeled the phosphorus, and it actually injects the DNA into the bacteria, and you get a radio-labeled bacteria. But when you radio-label this protein coat on the outside, nothing becomes radioactive because it doesn't inject the radioactive coat onto there. Okay, next we're going to talk about Chargaff, just very briefly. Okay, he figured out that there was an even amount of A and T. Okay, so for every adenine, there is a thymine. Chargaff's rule means that there will be an equal amount of A and T, and there will be an equal amount of G and C. So if you have 20% G, how much C will there be? Also 20%. Okay, then how much A and T will there be? If you have 20% G, you will have 20% C, so that's 40%. Now you have 60 over here, so this will be 30 and 30. Does everybody get that math? Are we all mathing? Are we all doing the same math? Same thing. A equals T, G equals C. That's where this rule comes from. It's called Chargaff's rule. And it also is purines bind with pyrimidines. And you will see how this helped Watson and Crick figure out the structure of DNA. And here's some of his data from, look at all the things he looked at. Humans, fruit flies, wheat, fungus, rats, E. coli. Does not matter. Doesn't matter. I mean, as it turns out, viruses, because they're always weird. There's always about an equal amount of A and T and almost an equal amount of G and C. See how that math works? That is Chargaff's rule. So DNA was first isolated from the pus, right? Then we went to Fred. They figured out that was the nucleon. They isolated the nucleus. Then Griffith, looking at Streptococcus, said, hey, something is transforming. This is crazy. They discovered DNA, but didn't know what it was, called it the transforming principle. Then Avery, McLeod, and McCarty showed that DNA is required for transformation, and Hershey and Chase proved it conclusively with the radioactivity. And then Chargaff again found the ratio of A to T and C to G. Now, this is the end of the video, so pause it here and see, can you explain what the transformation of DNA is? It's genetic engineering, but it's what happens automatically. The DNA automatically does that. We sort of hijacked its ability to genetically engineer. Describe the key experiments. We're going to go over these in lecture and explain Chargaff's rules.